So next is the chapter 2, biological classification. Chapter 2, biology classification. So the common introduction for biology classification and plant kingdom I already given to you in the lesson plant kingdom. Father of biology is Aristotle. He was considered as father of biology. He was the earliest to attempt a more scientific basis for classification. Means he was considered as father of biology, and he was the earliest to attempt more scientific basis for classification, because we know there were no picture about the classification of living organisms on that occasion. He simply classified the morphological level of organisms, and the plants are classified into trees, shrubs and trees. Trees, shrubs and herbs. Simple classification through the morphological level or morphological attempt. And plants are classified into trees, shrubs, herbs, and animals into red-blooded and non-red-blooded animals. Two categories. On the very beginning, so at the time when the Aristotle was considered as father of biology, so biology, study of bio, life, on that occasion, his time, the simplest classification. Plants are classified into trees, shrubs, herbs, red-blooded animals and non-red-blooded animals. Thus, the living organism, plants and animals. These are the living organisms, plants and animals classified in this manner by Aristotle. Later, advanced studies after Aristotle, the Carl Linnaeus classified the living system into two kingdoms. They are called plant kingdom, kingdom Plantae and Animalia. Kingdom Plantae and kingdom Animalia. Carl Linnaeus classified the living organisms into two categories or two kingdoms. namely kingdom Plantae and kingdom Animalia. It includes the plants. It includes the animals. This is the two kingdom classification. This system of classification did not distinguish between prokaryotes and eukaryotes, unicellular and multicellular, photosynthetic or non-photosynthetic, means autotrophic or non-autotrophic or heterotrophic. So the kingdom Plantae and kingdom Animalia, the two system of classification or two kingdom classification made by Carl Linnaeus did not distinguish between these three groups of categories, namely prokaryotes or eukaryotes. Prokaryote means a cell without a true nucleus. Eukaryotes, cell with a true nucleus. Such differentiation is not at all been present in this group of classification made by Carl Linnaeus. Unicellular or multicellular, the body or thallus of the plants or animals may either provide with a unicellular or more than one cell, unicellular or multicellular. Such classification is not at all included, and that means not at all distinguished. So generally he called plant kingdom and animal kingdom Plantae and Animalia. Unicellular or multicellular, then photosynthetic, that means autotrophic, those organisms which can produce their own food by themselves, autotrophic, or photosynthetic, and non-photosynthetic, having no production of food by them, or heterotrophic, they are dependent organisms. And so we can see the Carl Linnaeus two kingdom classification includes kingdom Plantae and kingdom Animalia. Kingdom Plantae includes of all of the plants and all of the animals. And here it did not distinguish between prokaryotes or eukaryotes, unicellular or multicellular, photosynthetic or non-photosynthetic. Later, in 1969, R.H. Whittaker classified the living organisms into 5 groups. So we know collectively called 5 kingdom classification. Five kingdom classification. The 5 kingdoms are kingdom Monera, kingdom Protista, kingdom Fungi, and kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. Kingdom Protista is the kingdom of prokaryotes. This is kingdom of unicellular eukaryotes. This is kingdom of multicellular heterotrophs. Kingdom of multicellular autotrophs. Kingdom of multicellular consumers. So, R.H. Whittaker proposed 5 kingdom classification in 1969. So, the 5 kingdoms are Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Kingdom Monera includes Kingdom of Prokaryotes. Kingdom Protista includes Kingdom of unicellular eukaryotes, cell with nucleus. Kingdom Fungi includes Kingdom of multicellular decomposers, or we can say the heterotrophs. Decomposers are all heterotrophs. Some are rarely, some are autotrophs. See, most probably they are heterotrophs. Kingdom Plantae includes Kingdom of multicellular autotrophs. Kingdom Animalia, actually the kingdom of multicellular consumers. And so this is based on the and then various criteria will be there. The main criteria of this classification is actually main criteria of this classification. A, the cellular structure. B, thallus organization. C, mode of nutrition. D, mode of reproduction. E, the phylogeny classification. So these are the 5 important criteria of classification of living organisms made by R.H. Whittaker. Cellular structure, thallus organization, mode of nutrition, mode of reproduction, and phylogenetic relationship. Relationship of organisms. See, once again, please listen. The different criteria, especially main criteria of five kingdom classifications, are the following: A. Cellular structure. B. Thallus organization. C. Mode of nutrition. D. Mode of reproduction. And E. Phylogenetic relationship. That means phylogeny, that you know. In the plant kingdom lesson, you know, already given the different kinds of classification in detail. And so phylogeny is actually the evolutionary relationship of organisms. Phylogeny. The term phylogeny is the evolutionary relationship of organisms. Relationship of organisms. That is phylogeny. So phylogeny or phylogeny relationship also needed for the classification of living organisms into five kingdoms by R.H. Whittaker. Next is Kingdom Monera. First is Kingdom Monera. Kingdom Monera includes prokaryotic organisms. It includes the primitive organisms. It includes the primitive organisms. It involves microorganisms. Mainly bacteria. So you know, Kingdom Monera includes prokaryotic organisms. Prokaryotic means an organism without two nucleus, or a cell without two nucleus is called prokaryote. It includes the primitive organisms. See, Archaebacteria, the bacteria which is seen very beginning of the origin of the earth. So you know, then Kingdom Monera involves microorganisms, mainly bacteria. Bacteria are unicellular organisms. Unicellular organisms. They are seen everywhere, wherever life is possible. Bacteria are unicellular organisms. They are seen everywhere, wherever life is possible. That is, they are omnipresent. That is, they are seen in water, air, soil, in and out of the body of plants and animals. Seen in water, air, soil, in and out of the body of plants and animals. And they are seen even in high temperature. Snow, etc. And the bacteria, the plant or animal, we can't say whether it is plant or animal, but formerly they are included as plant group because some autotrophic photosynthetic bacteria will be there. Anyway, the organism bacteria are seen everywhere wherever life is possible. That means they are cosmopolitan in occurrence. Cosmopolitan in occurrence. Cosmopolitan occurrence. Then that is the occurrence of bacteria. Then shape of bacteria. Bacteria are variously shaped. Bacteria are variously shaped. Based on their shape, they are classified into Bacillus, bacteria, which are rod-shaped in appearance. Coccus bacteria, Bacilli cocci, coccus bacteria. Sphere-shaped or spherical. Vibrio, comma shaped. Spirillum, sphere shaped. No, spirillum, spiral shaped. See, rod shaped bacteria. This is rod-shaped bacteria. Coccus bacteria. They are sphere-shaped, spherical in appearance. No locomotive organs like flagellum. They are non-motile and seen colonial except a monococcus. Monococcus bacteria contains only a single coccus, one. Bicoccus or dicoccus, that is two bacteria will be there. But most probably they are colonial. So non-motile and colonial. Non-motile and colonial. Bacterial. So these are Vibrios, comma-shaped bacteria. So the punctuation mark comma-like in appearance. Then Spirillum. So this is Spirillum bacteria. These are the different kinds of bacteria based on their shape. See, bacteria are variously shaped. Based on their shape, bacteria are classified into A. Bacillus bacteria, which are rod shaped. Coccus bacteria, sphere shaped or spherical in appearance. They are great to form a colony, and moreover colonial and non-motile, having no locomotive organs like flagella. Then Vibrios, that is Vibrae, that is comma-shaped bacteria. Spirillum, Spirillum, spiral, spring-shaped bacteria. They all possess locomotive organs like flagella, except the colonial bacteria like Coccus bacteria. See, structure of bacteria is very simple. Structure of bacteria is very, very simple. This is a simple bacterium having cell wall, not made of cellulose, but protein and glycogen are the components of cell wall. See, then this is plasma membrane. Which has numerous infoldings, and they are called mesosomes. They carry the enzymes. And then cytoplasm or protoplasm, which includes a circular DNA. The genetic material. And these are the locomotive organs called flagella. A rod-shaped bacteria having locomotory organs like flagella present, So most of the bacteria are flagellated, hence they are locomotory or motile. So the motility is due to the presence of these flagella, hair-like outdoors bearing from the cytoplasm, especially from the peripheral region of the cell. We can see cells are prokaryotic, having no true nucleus. Instead of that, a circular DNA present, which is considered as the genetic material. And very rarely, in addition to this, there is another circular DNA which is called plasmid, also present, that you can see later, not given in detail in our textbook. And then these bacteria has extensive metabolism, and these bacteria possess the extensive metabolism. So we know their nutrition. The bacteria are, most of the bacteria are heterotrophic. They cannot synthesize their own food by themselves. Hence, they are dependent organisms. They are free living. Some are autotrophic. They may be photosynthetic autotrophic. And chemosynthetic autotrophic. Photosynthetic autotrophic, purple bacteria, then chemosynthetic sulfur bacteria, etc. Then autotrophic or photosynthetic autotrophic. Photo or light is used for synthesis of food. They possess some photosynthetic pigments. Then example, purple bacteria, green bacteria, etc. you know. Then chemical substance for producing organic foods. Such bacteria are called chemosynthetic autotrophic bacteria. Such autotrophic bacteria will be there. See, heterotrophic bacteria are free living. They are depending. Free living means they are seen freely in the habitat. And some are symbiotic. Symbiotic. Say for example, E. coli, Escherichia coli, seen inside the intestine of man as a symbiotic bacteria. It is not at all harmful to us and beneficial to us also. Some are parasitic. Parasitic may be pathogenic. Disease causing. They secrete certain enzymes which will be harmful for the host. Ectoparasite and endoparasite may be there. So anyway, most probably bacteria are heterotrophic. They cannot prepare their food by themselves. They are free living in the habitat and do decomposition. So they yield the nourishment as well as the energy by decomposing the organic compounds are present in the habitats. They are autotrophic. They can prepare their food by themselves, and hence some are autotrophic, and they may be of two types: photosynthetic autotrophic and chemosynthetic autotrophic. In photosynthetic autotrophic bacteria, some pigments will be there which can absorb light energy to do or to synthesize their food through photosynthesis or light. Then chemosynthetic autotrophic bacteria, which are autotrophic because they can synthesize by utilizing light energy or chemical energy or chemical substances. Then some are symbiotic. They depend on other organisms. Example, E. coli in the intestine of man. Some are parasitic, may cause diseases. So the Vibrio bacteria causing leprosy. So like that, disease causing bacteria, they may be endoparasitic or ectoparasitic. And you know, the blight disease of plants caused by bacteria, citrus canker disease. Say pathogenic means disease causing, pathogenic disease causing bacteria. And so these are all about the nutrition of bacteria. Then bacteria reproduces, their reproduction. The reproduction of bacteria is mainly fission, binary fission. Asexually by fission or binary fission. Sexually by genetic recombination. That is transformation. Transduction. Transformation, transduction, and conjugation. Here, no a third progeny is formed or a zygote is not formed. The genetic material is transferring from one cell to another cell. That is called transformation. Transferring the genetic material through the virus is called transduction. And conjugation is when two bacteria of positive or negative strain come in contact with each other, they produce conjugating canal through which the genetic material is transferring from one cell to another. Not at all detailed in your textbook or in your syllabus, but you have to remember, please, only the sexual reproduction is by genetic recombination. In detail you can see in the next year. See, transformation, transduction, conjugation. In the second year lesson, you know, the genetic recombination very clearly seen. See, transformation, transduction, then conjugation. These are the process of genetic recombination, sexual reproduction bacteria. And that is about the reproduction in bacteria. Then, bacteria is classified into Archaebacteria and Eubacteria. First is Archaebacteria. Archaebacteria are considered as primitive bacteria. Archaebacteria are called as primitive bacteria, and their cell wall is very complex when compared with other bacteria. Their cell wall is very complex when compared with other bacteria, because this complexity give them great preventive or resistant capacity to the stress conditions. They can survive different kinds of stress from different kinds of stress conditions because of the peculiarity of their cell wall complexity. Then Archaebacteria is classified into three groups depending upon their habitats. So they are, first is halophiles. Seen in saline habitats. Then thermoacidophiles. Thermoacidophiles seen in hot spring and acid conditions. In hot spring and acid conditions. And C, methanogens. They are methane producing. Methane producing bacteria. Methane is a biogas which is highly inflammable, and so the Archaebacteria of three types: halophiles seen in the saline habitats, thermoacidophiles in high temperature and acid condition, then methanogens, they are methane producing methanogens, methane producing bacteria, and they are seen in the polluted conditions or polluted areas. They can produce methane as a biogas which is highly inflammable. Methane producing bacteria are called methanogens. So that is about Archaebacteria. That is about Archaebacteria. Next is Eubacteria. You know, some methanogens are also present in ruminant stomach of the ruminant animals like cows or buffaloes. So they are actually responsible to help to the digestion of cellulosic food that enter in the stomach. And these are all about it. Then Eubacteria. Eubacteria are the true bacteria. Eubacteria are the true bacteria. There are thousands of eubacteria. Eubacteria are true bacteria. There are thousands of eubacteria. See, cyanobacteria, a true bacteria which is unicellular or multicellular and filamentous. Filamentous cyanobacteria cyanobacteria are very like to blue-green alga. That contains chlorophyll A as green plants, as in green plants. Since chlorophyll A is there, they are photosynthetic also. So, you know, example, nostoc, anabina, etc. Nostoc is a colonial eubacteria. Nostoc. See, uni or multicellular and filamentous eubacteria present. If they are motile, they have flagella. If they are motile, they have flagellum as locomotory organs. The colony means aggregation of number of species. Colony of nostoc-like cyanobacteria are enclosed within a gelatinous matrix. Gelatinous matrix. But individual filaments is covered by a mucilage. Covered by mucilage. Formed of mucin, it is gelatin. Or remember please, the colony of nostoc-like cyanobacteria are enclosed within a gelatinous matrix. Gelatinous matrix. But individual filament is covered by mucilage. So we can see an individual filaments. So this is a no-stalk filament carrying spherical cells, vegetative cells. Among these vegetative cells, a number of enlarged cells called a heterocyst. These are vegetative cells. Fertile cell. And this is the no-stalk filament. Covered by mucilaginous. Mucilaginous covering of the filament, but an entire colony is covered, means a number of individuals covered by actually a gelatinous sheath, a gelatinous matrix, we can say. Within the gelatinous matrix, these individual filaments will be there, but individual filament is covered by a mucilaginous sheath. Remember, please. So the no-stalk like colonies are seen in polluted water as a bloom, a colony. Especially in the polluted water, it can be very clearly seen. Aggregate mass, cenobium, we can say. Cenobium, C-O-E-N-O-B-I-U-M. Cenobium means colonial nature, colonial habitat and cenobial habitat, like this. And so, since chlorophyll A present, no-stalk is autotrophic. See, photosynthesis. They can do photosynthesis. The autotrophic cyanobacterium. No-stalk. Autotrophic cyanobacterium. No-stalk. But chemosynthetic autotrophic bacteria will be, cyanobacteria will be there. Chemosynthetic autotrophic bacteria oxidize inorganic substances like it is important, substances like nitrate and ammonia and use release energy for their ATP production. See, chemosynthetic Eubacteria or cyanobacteria. No-stalk is a Eubacteria. chemosynthetic eubacteria oxidize inorganic substances, not organic, inorganic substances like nitrate and ammonia and release energy for their ATP production. So chemical substance is used for the synthesis of their foods according to chemosynthetic autotrophic bacteria. Say once again, chemosynthetic autotrophic bacteria oxidize inorganic substance like nitrate and ammonia and release the energy during oxidation for their ATP production. Thus energy can be utilized in the form of ATP, a chemical substance, and this chemical energy is used for their foods. Moreover, these bacteria help in the recycling of nutrients like nitrogen, phosphorus, iron and sulfur. The chemosynthetic autotrophic bacteria oxidize inorganic substances like nitrate and ammonia and release energy for their ATP production. At the same time, they help in the recycling of nutrients like nitrogen, phosphorus, iron and sulfur. These are general account of Eubacteria or cyanobacteria. Then the autotrophic bacteria may be helpful to man or other organisms and harmful to man, animals, pets, plants, etc. Say we know the economically they are beneficial to us. So generally we can say bacteria are our friends and foes. General accounts. When they are beneficial to us, they are our friends. Say for example, curling of milk, then production of antibiotics, curling of milk, which are beneficial to us. The production of ethyl alcohol, then preparation of the thein, alkaloids like thein will be coming out by the activity of certain enzymes of bacteria. Caffeine from coffee, they are all releasing out or coming out from the cell due to the activity of these enzymes. These enzymes rupturing the cellulosic cell wall and help to them the release of alkaloids from the cells. Cocoa, preparation of cocoa, then tea, coffee, etc. Bacteria are essential. So fiber from the husk of the coconut tree, releasing the fiber by tanning process, aquatic bacteria are needed. Then so these are some beneficial activities. So digestion is completed, cellulosic digestion is completed in man or herbivorous animals. So formerly I told you the methanogens that are present inside the ruminant stomach of buffaloes, cows, etc., like herbivorous animals, you know. And even in man, the cellulose is not at all digested by the enzymes of man. So we take the cellulosic foods. So here in the digestion of cellulose, the Escherichia coli, the symbiotic bacteria in the intestine of man, help to complete the digestion. So anyway, you have to remember the free living nitrogen fixing bacteria in the soil. See, nitrogen fixation by free living bacteria in the soil. Acetobacter, Nitrosomonas, Nitrococcus, Nitrobacter, so many bacteria. Just wanted to be one two. By free living bacteria and by nitrogen fixation by symbiotic bacteria. Symbiotic bacteria of roots of leguminous plants. Do nitrogen fixation. Then curdling of milk. Bacteria, Lactobacillus lactae like bacteria do curding, C-U-R-D-I-N-G, curding of milk to produce milk products like butter and cheese. So curding of milk, nitrogen fixation, then production of antibiotics, then preparation of some acids, preparation of fiber, curdling of milk by the production of milk acids, preparation of fiber, etc. are some beneficial aspects. But bacteria. So moreover, the most important process in the nature, the decomposition, to release the nutrients from organic compounds. Organic compounds are also nutrients, but absorbable and assimilable to the root system is the nutrients, mineral elements or mineral nutrients. So decomposition is in the soil to release the nutrients from organic compounds is one of the important role of bacteria, as they are the decomposers. So decomposition, preparation of fiber, preparation of some acids, production of antibiotics, curling of milk to produce milk products like butter and cheese, nitrogen fixation from in the soil by free living bacteria in the soil, or symbiotic bacteria of the roots of leguminous plants. So rhizobium like bacteria do nitrogen fixation. So they do the biological nitrogen fixation. The molecular nitrogen in the atmospheric air is absorbed by these nitrogen fixates and convert them into organic nitrogen. That is called biological nitrogen fixation. So generally we know the nitrogen fixation, curdling of milk, production of antibiotics, preparation of some acids, preparation of fiber, and moreover the most important role of this bacteria is decomposition, not to release the mineral elements or mineral nutrients from organic substances, and so they do the decomposition. These are all some beneficial aspects made by heterotrophic bacteria. Next is harmful effects. Some bacteria are harmful to us, harmful to human beings, animals, pets, and plants. And some bacteria are harmful to human beings, animals, pets, and plants. They may cause various diseases. Some are communicable. Various diseases. They cause various serious diseases, not only to man, but to the animals, domestic animals, or pets, and plants as a whole, we can say. And so, blight, diseases in plants, then leprosy, tuberculosis, tetanus, cholera, leprosy, tuberculosis, gonorrhea, tetanus, cholera, etc. are some animal diseases, especially in man. And these are all then you know, like canker disease. Canker, leprosy, tuberculosis, tetanus, cholera, gonorrhea, citrus canker, blights. These are plant diseases. These are anthrax, animal diseases. So, Hence they are called pathogenic. So the disease causing. Pathogenic means disease causing. Blight, canker disease or citrus canker disease, as plant diseases, leprosy, tuberculosis, tetanus, cholera, etc. are animal diseases. So generally we can say they possess certain harmful effects. So we know bacteria are our friends and foes generally. When they are beneficial to us, they are our friends. When they are harmful to us, they are our enemies. It means they may cause various serious diseases, not only in man but or other animals and in plants also. Hence they are harmful to us. They may destruct the crops, farm animals, farm animals, crops, plantations, etc. So different kinds of diseases both in plants and animals caused by the bacteria. And so these are all about general account of bacteria. Next is mycoplasma. They are the smallest living organisms. They lack cell wall. They can survive without oxygen. And they can survive without oxygen, and they may cause diseases both in plants and animals. Please. Mycoplasma are the smallest living organisms. They lack cell wall. They can survive without oxygen, and they may cause diseases both in plants and animals. And so these are all about mycoplasma. The points to remember. Next is Protista, kingdom Protista. You have to write down the flowchart. Kingdom Protista. Kingdom Protista involves unicellular eukaryotic organisms. Unicellular eukaryotic organisms. As it is eukaryotic, true nucleus present. Then membrane bounded cell organelles present. They are unicellular. Cell has a cell wall in some organisms, but cell boundary is there. Then eukaryotic, true nuclear contents separates outside the cytoplasm by means of a well-defined nuclear membrane. So nuclear membrane encloses all the nuclear contents. So well-defined nucleus present, membrane-bounded cell organelles, living inclusions, those which have biological functions or biological specialties will be there. Such living inclusions are also known as the cell organelles. So true nucleus present, membrane-bounded cell organelles present, characteristics of eukaryotes. Unicellular eukaryotic organisms are included in this group, kingdom Protista. So they are seen both in marine water and fresh water. Some are photosynthetic, some are heterotrophic, and some are autotrophic. Photosynthetic means autotrophic. Kingdom Protista is subdivided into Chrysophytes, Dinoflagellates, Euglenoids, slime molds, and protozoans. So by heart the names: Chrysophytes, Dinoflagellates, Euglenoids, slime molds, then protozoans. Protozoans are again subdivided into amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. First is Chrysophytes. Chrysophytes includes golden brown alga diatoms, golden alga, desmids, etc. Golden brown alga diatoms, golden alga desmids. They are unicellular, eukaryotics, seen usually in fresh water and marine water. In fresh water and marine water, that is habitats, and they float passively by the water currents. Passively by the water currents. Non-flagellated, cell wall contains silica, in addition to cellulose. The cell wall contains two house or shells. Cells are uninucleated. Cells are uninucleated. silicified cell wall is hard and indestructible. Diatoms are unicellular organisms. Cells are eukaryotics. They are usually seen in fresh water and marine water. They float passively by the water current. They are non-flagellated. Cell wall contains silica in addition to the cellulose. The cell wall contains two shells, seen just like soapbox. I can see one is smaller and other is larger. Cells are uninucleated. Silicified cell wall is very hard and indestructible. The cell remnants of billion years. Cell remnants of billion years accumulate to form diatomaceous earth. Diatomaceous earth. Since they are gritty, used for filtering syrup, since diatomaceous earth is gritty, it is used for polishing and the filtration of syrups and soaps. Syrups and oils. Syrups and oils. See, assemblies. This is not given in your textbook, the structure. A diatom, two shells, hard shells. One is small and other is large, which is overlapping the just like the soapbox, smaller one and larger one. Hypotheca, epitheca, name is not given. This is epitheca, smaller shell. Larger shell which overlaps the smaller one, just like the soapbox, closed soapbox, and their side view. This is cellulosic cell wall, and this is equipped with silica in addition to cellulose. Cellulose is there, and in addition to the cellulose, silicious nucleus is there. This is nucleus, large. This is small valve. Large valve is epitheca. Smaller one is hypotheca. Epitheca overlaps the hypotheca. Then centrally you can see nucleus. This is silicified cell wall. Billions of silicified cell wall. Cell remnants means silicified cell wall of billion years accumulate to form diatomaceous earth in the diatomaceous earth deep in the water. And since the diatomaceous earth is very gritty, it is used for polishing the metals or wood, etc., and used for filtration of syrups and oils and syrups, which are filtering with the help of this earth. And so this is general account about that. Moreover, you have to remember, please, since they are photosynthetic, they are the chief diatoms are the chief primary producers in the oceans. Diatoms are the chief primary producers in the oceans. And they are considered as the jewels of oceans because of their attractive color as well as the shape, structure, texture, etc. See, diatoms are considered as jewels of oceans. And that is all about Chrysophytes. Chrysophytes involves golden brown alga diatoms and golden alga desmids. They are unicellular eukaryotics, as it is Protista. They are seen both in fresh water and marine water. Then they float passively. The diatoms are generally seen in marine water. They float passively by the water current because of the absence of any locomotory organs. They are non-flagellated. Cell wall contains silica in addition to cellulose. Cell wall is there. In addition to that, silica is also present. The cell wall contains two halves or shells made of cellulose and silica. Cells are uninuclear, having only a single cell present in them. Silicified silica contained cell wall is hard and indestructible. The cell remnants of billion years accumulate to form diatomaceous earth deep in the sea. Since diatomaceous earth is gritty, it is used for polishing and the filtration of syrups and oils. So just imagine this is a diatom. Two halves are there as the cell wall. One is smaller, that is hypotheca, hypo below the epitheca. Then other is epitheca, hypotheca, just like the soap box, olden soap box, closing and opening. See, the cell wall contains cellulose and silica. Silicified cell wall is this margin. Silica deposition is indicating the lines. And centrally, you know, a nucleus. The nucleus, double nucleus now, single nucleus, small valve, large valve, that is hypotheca, epitheca, nucleus, silicified cell wall. Single nucleus is there. I wrote double. I wrote 2 times nucleus. No, 2 nuclei present. Nucleus. Clear? Okay. Another chief point. Important point. Diatoms are the chief primary producers in the oceans. Some red algae also perform the function of producers, chief primary producers, and they considered as jewels of oceans. And so these are all about a general account of Protista diatoms or Protista Chrysophytes. Protista Chrysophytes. Next is Dinoflagellates. They are photosynthetic marine Protista. Diatoms are also photosynthetic Protista. They appear yellow, green, blue, brown or red color depending on the prominent pigment. They appear yellow when yellow color is prominent. If green dinoflagellates. Most commonly, red colored, and that is due to the prominent pigment red color. The cell wall has stiff cellulosic plates. Cellulosic plates will be there. In Chrysophytes, cellulosic cell wall present. They are also photosynthetic. Here also, this is exclusively marine. Chrysophytes are seen both in freshwater and marine water, especially estuaries. Then, cell wall has stiff cellulosic plates, but here in Chrysophytes, the 2 overlapping shells or halves there. They are biflagellated. One is below in longitudinal, one is longitudinal and the other is transversal. So listen, a dinoflagellate. Suppose stiff cellulosic plates. make a furrow among them, two furrows or grooves. Stiff cellulosic plates, cyclops of the, so you know, see, this is a this is goniolates. This is goniolates. So listen, the cell wall is stiff cellulosic plate. They are biflagellate, two flagella will be there. Transverse flagellum seen on the furrows, transverse furrow. Then vertical or longitudinal furrow is there. There also elongated flagella help in twisting inside the water, which help to the movement of the organism. One is enclosed in the transverse furrows. When they are become inverted, the transverse flagellum becomes vertical flagella. Vertically seen, flagella remain there. Red dinoflagellates are very often because red color is very prominent. Example, gonialax. This is gonialax. They undergo rapid multiplication to make red tides. See, at the sea surface or seawater surface, red tides will be there due to the abundant accumulation of young ones of these red dinoflagellates. There, they secrete some poisonous substances. And it kills the fishes and other aquatic animals. Fishes and aquatic organisms. See, listen. Dinoflagellates are photosynthetic marine Protista. They are usually seen in marine water, exclusively marine. They appear various colors like yellow, green, blue, brown, red, and appear red depending on the prominent pigments. Yes. The cell wall has stiff cellulosic plates which make longitudinal and transverse furrows or grooves. They are biflagellated, having a longitudinal and transversal filaments or filament structure called flagellum will be there. Since two biflagellated, two flagella are there. Red dinoflagellates are very often in the sea. And example, gonialax. See, they undergo rapid multiplication to make red tide. They secrete some poisonous substances, the young ones, and it kills the fishes and other aquatic organisms. So that is about general account of dinoflagellates. Next is euglenoids. That is on the next class. Okay, see you.